It is my great pleasure to welcome today in Kraków Professor Walter Gilbert, the physicist, chemist, molecular biologist, who won the Nobel Prize in Chemistry in 1980. Professor Gilbert is not only the scientist, he's also a successful businessman and a great artist. It is not science that brings you to Kraków today, but art. Last week, we had the pleasure to participate in the grand opening of your exhibition of the digital art that you do. Are there any similarities between art and science?
There's some very striking similarities in the creative impulse. Both art and science share a very strong creative drive. In science, that creative drive is to find something true and new about the world. In art, that creative drive is to create something new again, but something that has an emotional impact on the viewer. And so I found it in certain ways possible for me to move from one to the other, although the art that I do and the science that I did are not really related. The science was an experimental science, trying to find out new things about biology. The art is an effort to create aesthetically pleasing images. I use a computer, but I use it simply as a tool, like a painter's palette. It provides me with colors, it enables me to do things, but it doesn't involve the use of the computer as a mathematical tool.
In the summer of 1960 at the Harvard University, James Watson, who together with Crick was awarded the Nobel Prize for the discovery of the DNA molecule structure, invited you to join his research team that was trying to identify messenger RNA in order to discover how proteins are synthesized. Why did Watson invite a physicist to join his team, and what was so exciting about their research that made you shift your interest from theoretical physics to molecular biology?
That's a big question. I met Jim Watson when I was a graduate student in Cambridge, England, where I took a degree in theoretical physics in that country called mathematics. Jim and I became friends, and when we came back to Harvard, I still as a graduate student and he as assistant professor, we remained friends for the next several years. A few years after I became an assistant professor of physics, Jim could tell me or say to me, something very exciting is happening in the lab. And I went over and visited, talked to him and his colleagues at that time, and decided that yes, it was exciting, and physics was actually going through a dull moment, and so I joined in doing the experiments. And that summer and fall, Jim and I and another colleague, Francois Gros, worked intensively doing experiments about messenger RNA. I found that I had a natural ability to do experiments, even though I had been a pure theorist in physics. But experiments are dominated by the concept, the ideas, so that there's certain ways I could take over the conceptual ability I had in physics and apply it in biology. But it was a very amusing period because I knew nothing actually about the biology. And so I'd simply have to ask people questions all the time. And I would bother the students in the lab by asking them questions and gradually learn enough to fully participate.
Your research in the field of molecular biology led you to the discovery of the rapid DNA sequencing method, together with Frederick Sanger. In 1980, you were awarded the Nobel Prize for this discovery. Why is the sequence of DNA important, and what was the impact of your discovery?
Fred Sanger in England and I and my colleague Alan Maxim in the United States discovered what we call rapid ways of working on the DNA sequence. The order of chemical groups along the DNA carries all the information that's passed from parent to offspring. And so the underlying problem, one of the underlying problems in biology is how do we understand all that information, the genes? I had worked out a small piece of DNA, 20 units long, and it took us 2 years. Essentially 1 unit, these units are called bases, 1 unit a month. A typical gene is 1,000 to 10,000 units, and you would realize at that speed, you would never be able to understand even the simplest gene. In 1976, I discovered in the States, and Fred in England discovered extremely rapid ways of working out the DNA. And so the problem went from 1 chemical unit a month to hundreds or thousands in an afternoon. That process, we published it we announced it immediately. People began sequencing genes all around the world. And the DNA sequencing has increased a factor of about 1 million in rate since that time. There are now machines available that in an afternoon work out 1 billion bases of DNA rather than the hundreds or thousands we did in the first time. This discovery had totally transformed biology. The problem in biology, one of the basic problems, is what are the genes and how do they work? We could, by deciphering the DNA, at least have a list of all the genes. The effort around the world went from a gene a year to a gene a month to a gene a day to thousands of genes immediately. The ability to do this rapid DNA sequencing has led to things like the Human Genome Project, that in fact the entire genome of a human being, and by now several thousand human beings, have been deciphered. And that knowledge is, or that ability, or say that the knowledge that we get that way, which is essentially a list of all human genes, leads us to attempt to understand those genes. I used to argue that the Human Genome Program represented a way of working out the sequence of all the DNA in a few years, but it would take 100 years to actually understand how it all works. And that's one of the problems that we have in biology today. The consequence of being able to see the operation of genes means that we can, at one level, identify the genetic causes of many diseases. At another level, it means we can begin to understand in detail how the organism develops from an egg into an entire person. So medical science has been transformed, by our ability to look at the DNA sequence. The way in which we look for new drugs today is based on our ability to identify a gene and a protein the gene makes and see that we wish to change the activity of that gene. We wish to make less of that protein or more of it or inhibit its activity. And so many diseases are attacked by trying to identify chemical compounds that will either interfere with or activate a protein. So that the lead way in which we try to find new drugs is through knowledge of the genes and their products. The way in which we try to find drugs for cancer is by looking at the particular genetic defects that have occurred in the cancer cell and trying then to say, well, those defects lead the cell to grow in certain ways, and we can then try to interfere with those ways specifically. In medicine, the ultimate application of this is we'll call personalized medicine. That we move to realize that each person has a unique constellation of biochemical attributes directed by the specific way their genes function. And one really wants to design medicines specifically for the individual. That work well in that individual, that correct the particular problems that individual has. And the entire thrust of, a large part of the thrust of medicine today is going in more and more in this personalized medicine direction.
Many scientists from Harvard University have been Nobel Prize laureates. There are also many who have significantly contributed to the progress of their respective disciplines without getting the Nobel Prize. Many Polish universities try to implement programs to support innovation. What is so special about Harvard that allows scientists to grow, to discover, to create, and to innovate?
There are many, many good universities in the world. Harvard is actually a small university. The basic attitude, they're very strong attitudes at a place like Harvard or Caltech or the other major universities in America that try to permit people to have time, enough time to do research, and enough support. I mean, some of the question is level of financial support that's available. A lot of the question turns on the degree to which people are not spending all of their time teaching, but are in fact able to spend time creating new knowledge, and the nature of having a group of people around them that stimulates them in that creation of new knowledge. It's very hard to function in isolation, let's say, from the thrust of world science. And so that interaction is very, very important. In some fields, that interaction is occurring more and more on the web. Mathematics, that field, that occurs more and more as people communicate around the world. In laboratory fields, you need a sufficient number of people, you need the laboratory equipment, and you need the inspiration of other people.
You are a scientist, an artist, and a businessman. You co-founded biotech companies such as Biogen and Myriad Genetics. You are also a general partner at BioVentures, a venture capital firm with a really impressive portfolio of biotech companies. How have you been able to combine these three parts of your professional life so successfully?
I wish I had a convenient answer to that I could pass on to somebody else. There's several themes. One, in my life, I've changed the fields in which I worked quite strikingly. I was originally a theoretical physicist. I then became a completely experimental biologist. I then left and ran a company. And now I'm spending most of my time doing art. I found that it's possible for somebody to change and to change what they're doing. One of the great dangers in the world is that people feel that they must do what they've been trained to do, that they've been educated to do a certain thing, and therefore that's going to be the form of their life. They don't realize that life is long and one can change completely what one's doing. I could change from physics to biology by actually having gone through the full training in theoretical physics and having written a PhD. And I could realize that picking up a new science was a matter of learning specific techniques. It wasn't a matter of learning how to think about science again. And in going into business, I found that, again, I had the drive to do it. I had the confidence to do it. I had enough ability to do it. And so I went ahead and did it. The same is true in the art. In some ways, there's an underlying creative drive in all of these that I find very natural.
You have been a part of some of the most exciting scientific discoveries of the 20th and 21st centuries. How do you envision the future of science?
A very exciting one. I think the future of science will continue to astound us. The element that drives one as a creative scientist is, in fact, the search for something new. We're constantly curious. We can barely bring ourselves to talk about the old work, but we have to in order to teach. But the creative work in the laboratory is that search for the new fundamental finding. The scientific world is still filled with a plethora of questions. In biology, there are a series of practical questions, the medical questions, the cure for cancer, treatment for Alzheimer's, the gradual development of ways that support the body. There'll be very striking discoveries made in that. We have a list of all the genes, but we do not, in fact, know all the ways in which the genes interact, and we do not know how they're all controlled. There's an entirely exciting field of biology, which is still called neurobiology, and the fundamental questions there are questions about how the mind works, and questions like, how does memory really work? How do we recover a memory? What is the nature of consciousness? How does our brain really work in a fundamental way? These are questions which are still not really not understood, and there are great breakthroughs still to be made there. Astronomy is fantastic. Our vision of the universe has changed drastically in the last 10 years and changed drastically in the 25 years before that. So now we know that the matter we're made of represents only about 4% of what's there in the universe. And the rest is filled with dark matter and dark energy, and in ways that's being explored today in fantastic ways. And yet, discovery of what these things are is still in the future.
You are also known for coining the term the RNA world. What does it mean?
Yes, the RNA world is it's a vision of how life might have originated. We think we know, I should say, that life originated on Earth of the order of 3.5 billion to 4 billion years ago. That's the first time we see traces that are left in the record. We have a choice of either suggesting that life in the form of single cells originated here or originated somewhere else in the universe, but there's only 14 billion years for that to happen, and it is possible that life came in from outer space. It is generally thought that there's probably that life on Earth would have also been transferred on meteorites to Mars, so life on Mars would have been transferred on meteorites to Earth, but no one has yet proven anything like that. When one tries to think of what the first living forms might have been, a striking thought emerged that's called the RNA world. It's really the suggestion that although today DNA contains the genetic information, it's copied into another molecule called RNA, slightly different in chemistry, and that molecule is in turn used as a set of instructions to make a protein. That separation of DNA to RNA to protein sort of separates the notion of genetic material, the material that's inherited by the offspring, from the operational material, the proteins that are the enzymes that make chemical reactions happen. A discovery several years ago by Thomas Cech and others showed that RNA could also be enzymes. And that led me to suggest a notion of the RNA world, that maybe the first molecules that had to be assembled and could serve both as genetic material and as the enzyme that copied the genetic material. And so you'd have a world made up of essentially only RNA molecules serving both as the enzymes and as the instructions to make new enzymes. And then one could, following that conception, could see how DNA could be a later development, how the proteins could be made a later development. And there's a series of arguments that suggest that something like the RNA world may underlie the very first organisms that begin life on Earth.
Thank you so much. It's been an honor, sir.